Hello everyone, my name is Grace Hurst, and today I will be presenting photosynthesis but make it arsenic, the story behind Mono Lake's peculiar bacteria. This presentation is brought to you by BIO282 Lab. Let's begin with a rundown of today's presentation. We'll start with an analysis of our news article, which was published by the New York Times. We will then move on to our primary literature article, which was published by the American Association for the Advancement of Science, also known as Science Magazine. We will then compare the two and see exactly how well the news article covers the scientific literature and whether or not it is an accurate summary. Lastly, I will go over the credits, which will contain the sources for the two articles and the acknowledgments. The title of our news article is In Lake, Photosynthesis Relies on Arsenic. This article was written by Henry Fountain. The news article jumps right in with a description of the mysterious Mono Lake, a strange place with eerie spires of calcium carbonate along the shoreline and water that is extremely salty and alkaline. The author then goes on to describe the discovery of a new bacteria which uses arsenic instead of oxygen for photosynthesis. This new bacteria grows in pools on Paoha Island in Mono Lake. These pools are fed by hot spring water. The photosynthesis begins as usual using light to produce carbohydrates from carbon dioxide. But then, instead of using water to produce oxygen, the bacteria uses arsenite ions to produce arsenate ions. This new bacteria is similar to other bacteria that has been discovered that uses sulfide, nitrate, and ferrous ions as electron donors and are also anoxygenic or non-oxygen producing. But the Mono Lake bacteria is the first of its kind to photosynthesize using arsenite. Let's move on to our primary literature article. This article is titled Arsenic-3, also known as arsenite, fuels anoxygenic photosynthesis in hot spring biofilms from Mono Lake, California. Let's start by looking at a figure from the article. This figure shows two samples collected from a red pond and a green pond on Paoha Island. Both of these ponds contain high levels of arsenite and contain arsenate as well. The two ponds are compared to the surface water in Mono Lake, which contains no arsenite but a high level of arsenate, which is most likely produced by the bacteria that is being researched. Now let's move on to the science of it all. The green pond samples slowly oxidize arsenite to arsenate under anoxic or oxygen-free conditions in the light but not in the dark. This is because photosynthesis cannot commonly proceed without a source of light. The red pond sample oxidized arsenite as well, and subsequently injected arsenite was then also completely oxidized to arsenate. When tested in dark anoxic conditions, arsenite did not oxidize. The scientists then tested a strain of this arsenite-dependent bacteria found in the ponds named PHS1. This bacteria successfully grew in its usual light conditions when provided arsenite, but unsuccessfully grew when the light was taken away and also when the arsenite was taken away. To conclude the article, the scientists wrote, "Our discovery of anaerobic photosynthetic oxidation of arsenate adds an important new dimension to the arsenic cycle and highlights a previously unsuspected mechanism that may have been essential for establishing and maintaining the arsenic cycle on the ancient anoxic Earth." Now we are able to review the two pieces of writing. This is the final test. Does the news article correctly summarize the scientific findings? Let's find out. First, let's go over how the news article is accurate. To begin, the news article does describe the type of bacteria that was found in Mono Lake. It also describes the process and how the photosynthesis works for this bacteria. Finally, the article is very simple and does a great job of taking the intense and wordy scientific findings and displaying them in a simple manner. Now let's see how the news article is inaccurate. The good news is that it isn't. There are no inaccuracies seen in the news article, but there are a few flaws that could be improved upon. For example, the lack of details. The experimental methods that the scientists used to discover this bacteria were not included in the article. Although this article is suitable for a reader in a hurry, it may benefit from explaining how the scientists came to their conclusion. Finally, the credits. Shout out to BIO282 Lab, which is the class that this presentation was created for. Next, we have our citation for the news article used in today's presentation, as well as the citation for the primary literature article. Next, I would like to acknowledge Slides Go for the wonderful template used in this presentation. The final acknowledgement is myself, because I am the creator of this presentation. Thank you all for watching, and I hope that you learned something new today.